Welcome to our journey into the ancient oceans. Today, we're going to explore the superclass Agnatha, the jawless pioneers of the vertebrate world. Understanding these incredible survivors is like looking through a biological time machine, revealing the very first steps of vertebrate evolution and showing us what our earliest ancestors looked like. To begin, let us imagine swimming in an ancient ocean long before sharks or bony fish existed. You might encounter a bizarre eel-like creature, but as it gets closer, you notice it has no jaws to bite you, no scales for protection, and no paired fins to steer. Instead, it has a round, sucker-like mouth filled with tiny, sharp teeth, acting almost like a living underwater vacuum cleaner. These are the agnathans. Modern survivors, like lampreys and hagfishes, hold on to this primitive body plan because it has worked perfectly for hundreds of millions of years. Now that we have a picture of these creatures in our minds, let us look at their formal biological classification. Agnatha is a superclass of jawless fish within the phylum Chordata and subphylum Vertebrata. The absolute most defining characteristic of this entire group is the complete absence of hinged jaws. This sets them apart from all other vertebrates, which we call gnathostomes. Furthermore, agnathans completely lack paired appendages. This means they do not have the pectoral or pelvic fins that eventually evolved into the arms and legs of land animals. Moving inward from their lack of jaws and fins, their internal anatomy is equally fascinating. The internal skeleton of an agnathan is entirely cartilaginous. This means it is constructed of flexible cartilage rather than the hard, calcified bone found in more advanced vertebrates. Additionally, they retain a primitive rod-like support structure known as a notochord. While in most vertebrates this structure is replaced by the vertebral column during development, agnathans retain this notochord as their primary axial support throughout their entire adult lives. In addition to their unique skeleton, agnathans have distinct adaptations for breathing and protection. They possess a highly specialized respiratory system consisting of multiple pairs of gill pouches. Depending on the species, they typically have anywhere from 7 to 14 pairs of these pouches, which open directly to the outside aquatic environment. Furthermore, if you were to touch one, you would notice their skin is completely smooth and highly glandular. They are entirely devoid of the protective dermal scales that cover the bodies of most modern fishes, relying instead on thick mucus for protection. Understanding these anatomical traits leads us to why agnathans are so incredibly important to science. For evolutionary biologists, these creatures are a treasure trove of information because they represent the basal, or most primitive, group of living vertebrates. They serve as a living model, showing us exactly what our earliest ancestors looked like before the massive evolutionary invention of jaws. This transition to jaws was a turning point that allowed animals to become active, biting predators. By studying agnathans, we gain direct insights into the evolutionary origins of our own jaws, immune systems, and skeletal structures. Beyond their evolutionary importance, agnathans play significant roles in modern ecosystems and human economies. In the deep ocean, modern agnathans, like hagfishes, act as vital scavengers. They recycle essential nutrients by consuming dead and dying animals that fall to the ocean floor. On the other hand, lampreys are often parasitic and can have a massive economic impact on commercial fisheries. In regions like the Great Lakes, invasive lamprey populations have historically decimated populations of valuable fish. Studying their unique biology is absolutely necessary to develop strategies to control these invasive populations and protect commercial fisheries. Having discussed their ecological impact as parasites, how exactly do these jawless lampreys feed? They rely entirely on creating a powerful vacuum to suck blood and tissue fluids. We can model this biological mechanism using the Hagen-Poiseuille equation for fluid dynamics. In this formula, Q represents the volumetric flow rate, telling us how much fluid the lamprey ingests per second. Delta P is the suction pressure difference generated by the throat muscles. The variable R is the radius of the puncture wound. Eta is the dynamic viscosity or stickiness of the host's blood, and L is the length of the feeding tube. Notice that the radius is raised to the fourth power. This means if the mouth opening increases even slightly, the flow rate increases dramatically. Conversely, thicker blood with higher viscosity will decrease the flow rate. Let us apply this formula to a real-world scenario with a sea lamprey. We are given a suction pressure of 2,000 pascals, a radius of 0.002 meters, a tube length of 0.05 meters, and a blood viscosity of 0.004 pascal seconds. We substitute these values into our equation. First, we expand the radius to the fourth power, yielding a very small number, and multiply the constants in the denominator to find the total resistance factor. Next, we simplify the entire numerator, which represents the total unresisted flow potential. Finally, dividing the numerator by the resistance gives us our answer. The flow rate is 6.28 times 10 to the negative fifth cubic meters per second, or about 62.8 milliliters per second. This shows that despite lacking jaws, the lamprey's suction mechanism is incredibly efficient at draining vital fluids. Moving from feeding to breathing, agnathans rely on their multiple gill pouches to extract oxygen from water. We can understand this using Fick's law of diffusion. In this formula, J is the required oxygen diffusion rate for the animal to survive. A represents the total surface area of the gill pouches. D is the diffusion coefficient, which tells us how easily oxygen moves through water. Finally, dC over dx represents the concentration gradient of oxygen across the gill membrane. Conceptually, this formula tells us that the total amount of oxygen absorbed depends directly on how large the gill surface area is and how strong the concentration difference is between the water and the blood. If the surface area A increases, the diffusion rate J increases proportionally, which is why these creatures need so many gill pouches. Let us see this in action by calculating the required gill surface area for a hagfish. We are given a required oxygen diffusion rate, J, of 5 times 10 to the negative 6 moles per second. The diffusion coefficient, D, is 2 times 10 to the negative 9, and the concentration gradient is 500. Because we need to find the surface area, we first algebraically rearrange Fick's law to isolate A. Next, we substitute our given values into the rearranged equation. The numerator is the target oxygen flow rate, and the denominator represents the physical diffusion capacity. We simplify the denominator, perfectly aligning the powers of 10. Dividing these gives us a final answer of 5 square meters. Physically, this massive required surface area explains exactly why agnathons possess such an extensive, highly folded network of up to 14 pairs of gill pouches. Now that we have explored the math and concepts, let us look at the actual anatomy of a jawless fish in this diagram. Starting at the far left, you can clearly see the round buccal funnel, or mouth, equipped with a rasping tongue, completely lacking any hinged jaws. Moving just behind the head, notice the row of 7 distinct circular openings. These are the external gill slits we just calculated the surface area for. Along the top, there's a simple dorsal fin for basic stability. However, look at the lower half of the body. The arrows explicitly point out the complete absence of pectoral and pelvic fins. This streamlined, primitive body plan is exactly what makes the agnathans so unique in the vertebrate world. We have covered a lot of ground today, so let us review our key takeaways. First, the superclass Agnatha represents the most primitive lineage of vertebrates alive today. They are fundamentally defined by their lack of hinged jaws and paired fins, relying instead on a cartilaginous skeleton and notochord. Second, instead of biting, they use specialized suction and rasping mechanisms to feed, which we successfully modeled using the Hagen-Poiseuille equation. Third, to survive without advanced pumping systems, they rely on multiple pairs of gill pouches to maximize oxygen diffusion, as proven by Fick's law. By studying these fascinating living fossils, you have gained invaluable insights into the early evolutionary steps that eventually led to all modern vertebrates. Great job completing this topic.